Welcome back, everyone. So we're going to get into Unit 2 this week. And this week covers just one chapter. So we're going to cover Chapter 3, which is the molecules of life. So this chapter is divided into basically 5 subsections. So we have where we're going to introduce what organic compounds are, as well as describe the 4 macromolecules of life. So the carbohydrates, the lipids, the proteins, and the nucleic acids, which basically make up you. So by the end of this particular lecture, you should be able to describe the importance of carbon to life's molecular diversity. You should also be able to describe the chemical groups that are important to life. So we'll talk about those functional groups. You should be able to explain how a cell can make a variety of large molecules from a small set of molecules. So we'll go through what type of reaction that involves, as well as define monosaccharides, disaccharides, and polysaccharides, which are carbohydrates, and explain their functions. And then finally, we're going to define lipids. So lipids being basically another type of macromolecule. Some examples being phospholipids and steroids, and explain their function. And then related to that, we're going to learn a little bit about trans fats and how they're formed in our food products. And you'll be able to describe the evidence that suggests that eating those trans fats is not nearly, not very healthy for you. So it's actually worse than consuming saturated fats. So by the end of this lecture, you should be able to write out all of these explanations. Okay. So let's start out by talking about molecular organization. So if you recall from chapter 1, we talked about the hierarchy of life, and so that lowest level of life's hierarchy being the atom. So in chapter 2, we learned all about atoms, and then how atoms, they can form together to form molecules. So molecules, those are compounds of 2 or more atoms held together by a chemical bond. Okay, so as we go through this particular chapter, you're going to learn about the 4 macromolecules of life. But before we get into those macromolecules, we need to understand how their structure, molecule structure, is related to their function. So we're going to learn more about those carbon-based molecules, and understand that just a small change in their structure can have big changes in the function of that molecule. So let's start out by learning about organic compounds. So anything that's organic is carbon-based. So remember from chapter 2, we talked about how carbon is basically, it has 6 protons, so that means it has 6 electrons. So the first 2 electrons fill the first shell, but then they have 4 electrons in their outermost shell. And what was that outermost shell called? The valence shell. So in their valence shell, they have 4 electrons. And if you recall from chapter 2, that second shell can hold as many as 8. So they want to be able to fill their outermost shell, but they can't do so by gaining or losing electrons in an ionic bond. So instead, they share those electrons. So they share electrons with up to 4 other atoms in what we call covalent bonds. And remember, covalent bonds, those are sharing those electrons. So it's a very strong bond. And here's an example of an organic compound. So this is methane. So methane, in this case, you can see has 1 carbon and 4 hydrogens. So each one of those hydrogens shares an electron with the carbon. So they are filling their outermost shells. So let's start out with an animation. We're going to learn about carbon and carbon skeletons.
The carbon backbones of organic molecules may vary in length and in location of single and double bonds. Molecules may be straight or branched and may even contain rings of atoms.
Okay, so just from that brief video, any of the dark color or almost black color balls there, those represent carbon atoms. Okay, so you can see how carbon atoms, they can be branched together in a more linear fashion. They can even have double bonds. They can form a more branched versus the linear, as well as have more of a ring structure. Oops, let me go back. So what are carbon skeletons? They're just chains of carbon atoms. So you can see several different examples here. We have carbon skeletons. Their length varies, okay? So we can have, 2 carbons held together. We can have 3 carbons. We have more than 3. So the carbon skeletons do vary in length, okay? And then as far as those bonds that are bonding the carbons, remember these are covalent bonds, and those bonds could be a single bond like shown in propane, or it could be a double bond shown in 1-butene. Okay, so we have a double bond between the first and second carbon in 1-butene, whereas in 2-butene we have the double bond between the second and third carbons. Okay, and these two particular molecules, by the way, they're called isomers. So isomers basically have the same chemical formula, meaning they have the same number of carbons and the same number of hydrogens, but they differ in structure. So you can see how the bonding is different in these two isomers. Okay. Here's another example of an isomer where we have branching. So butane, as we saw before, but in the double bonded version, butane is just a linear organic compound, whereas isobutane is branched. But if I added up all those carbons and hydrogens, they would be equal to each other. So this is another example of an isomer. Okay, so isomers have the same chemical formulas but different structures. Okay, so in this case we have a branching structure versus a linear structure. And then finally we saw in the video how carbons, they can form rings. Okay, so here is an example of a single ring structure, and that's just denoted by, well, in this case there's 6 rings, so a hexagon. So each one of these lines where they meet indicates a carbon, and then that's a single bond with another carbon. Whereas in benzene, we actually see a different shaped hexagon, and it still has 6 carbons, but you can see a lot less hydrogens, so we have double bonds forming between the carbons. Okay, so carbon skeletons, in the rings you might see abbreviated like that instead of including all of the carbons and the hydrogens. Okay. So we're going to watch an animation about the different types of isomers. So remember, isomers share the same chemical formula but different structures. So there's 3 main types: the structural isomers, the geometric isomers, and something called an enantiomer.
3 types of isomers are structural isomers, which differ in the covalent partnerships between their atoms; geometric isomers, which vary in arrangement of atoms around a double bond; enantiomers, or stereoisomers, which are molecules that are mirror images of each other, like left and right hands.
Okay, so you can see the difference between the 3. Okay, so structural isomers, they have the same chemical formula, but they're structured differently. So we have a more linear one here and a more branching one here. In a geometric isomer, they actually look exactly the same here, but depending on where those extra atoms are. Okay, in this case, this is called a trans isomer because it's on opposite sides, where this is a cis isomer. Cis means same, so these 2 atoms, whatever they are on the same side together. Trans, opposite. And then finally we have the enantiomers. So enantiomers, if I put a mirror right down the center, they're mirror images of each other. Okay, so they're, here's the gray, and then the red, the blue, and then the green. So they're mirror images of each other. Okay, now as I keep talking about, organic compounds, they their structure will help determine their function. So we're going to look at what are called functional groups, and they help us determine what exactly that molecule's function is. Okay, so we're going to look at the properties of each one of these functional groups. So again, we already know that the organic compounds, they depend on the size and the shape of the carbon backbone, but they also depend on those functional groups. Okay, so those are groups of atoms that are attached to the carbon skeleton. So here you have a carbon skeleton with an oxygen double bonded. Okay, here you have a carbon skeleton with an oxygen and a hydroxyl group double bond or bonded. So these are examples of functional groups. And when we're talking about each one of these functional groups, they tend to be polar. So if you recall polar, what does polar mean? Okay, they mean that we have that difference in electronegativity, and so because of that, we have one particular atom within the molecule kind of holding on to the electrons more than the other atoms attached, and so they have kind of an electronegative gradient forming. And another thing, because they are polar, they also tend to be hydrophilic. So what is that term hydrophilic? Hydro means water, philic means liking or loving. So hydrophilic means that these particular functional groups, they tend to be hydrophilic or water-loving. Okay, so we're going to take a closer look at these are basically 6 types of functional groups, and this is a figure from your table. So to get us started identifying the different functional groups, I'd like to do this checkpoint question. So you're going to use the table 3.2 to identify the chemical groups that do not contain carbon. Okay, so this is just related to the functional groups being the highlighted groups there. So which groups do not contain carbon? Well, the first one, hydroxyl group, that is doesn't contain carbon. Okay. So that one you can see attached to the carbon skeleton, but the functional group itself does not contain carbon. Carbonyl and carboxyl, they both sound like carbon, so they contain carbon. Amino groups, they do not contain carbon, so they have a nitrogen and 2 hydrogens attached. And then the phosphate group, phosphate contains phosphorus and some oxygen, so that one also does not contain carbon. And then finally we have our methyl group. So we talked about methane, where we have a carbon attached to 4 hydrogens, but we can also have a methyl group where we have a carbon attached to 3 hydrogens, but is also attached to the carbon skeleton. Right? So there's 3 out of 6 of these don't contain the carbon. So the hydroxyl groups, the amino groups, and the phosphate groups. Okay, so we're going to go through some examples of these functional groups. We'll go through the first 3. We'll talk about them though. Okay, so starting with the first one, we have the hydroxyl group. So if you look at the hydroxyl group, sorry it's a little fuzzy there, the hydroxyl group, we have a hydroxide attached. Remember, hydroxide is OH. And hydroxyls, they are also known as the alcohols. Okay, so in this case, we have ethanol. So ethanol is an example of a molecule that contains a hydroxyl group that is considered an alcohol. Another example would be an amino group. So amino groups, they have basically the nitrogen and 2 hydrogens attached. So those are known as the amines. So you might think of something like an amino acid. So amino acid, we'll learn more about when we in our next lecture when we talk about proteins. But amino acids, they contain an amino group as well as another functional group, which I'll go back and show you. the carboxyl group. Okay, so carboxyl groups, they contain an OH, like our hydroxyl group, but they also contain a double bond to an oxygen for that carbon. Okay, so we call, when it's just a carbon double bonded to oxygen without the hydroxyl, we call that a carbonyl group. So carbonyl group is just the carbons double bonded to the oxygen. But when we add that hydroxyl group, we get carboxyl. So think carbonyl just has the double bond to oxygen. Okay, like here is an example. But when we add a carbonyl group with an hydroxyl group, we get a carboxyl group. Okay, so we can see those are two examples of the carbon ones. So we talked about hydroxyl groups, carbonyl groups, carboxyls, and amino groups. And then the methyl group, that's just another one with the carbons with the 3 hydrogens attached. And then the final one we're going to talk about are the phosphate groups. So the phosphate groups, also known as the organic phosphates. So our organic phosphates, they have the carbon skeleton, or whatever the rest of the molecule is, but then they have an organic phosphate, and oxygen, and then a phosphorus with 3 oxygens attached. That is an organic phosphate. Okay. In this case, this is an organic molecule called ATP, adenosine triphosphate. So we have adenosine, which is actually a nucleotide related to nucleic acids. We'll talk about that in the next lecture. And then we have 3 phosphate groups attached. So adenosine tri means 3 phosphate. Now we keep talking about how our functional groups, they will determine the function of that particular molecule. So if we change that functional group, it can cause a big change in the function of that particular molecule. So for example, let's take a look at these two. These are steroids. We have testosterone and we have estradiol. Okay, you might know them as sex hormones. And if you look at the structure, okay, the carbon structure, which is these rings, they look very similar to each other. Okay, the only difference is the functional groups. Okay, so they have a methyl group and a hydroxyl group at the top, but then estradiol has a hydroxyl group over here, which testosterone does not. Instead, testosterone has, what is it called when you have a carbon double bonded to an oxygen? That's a carbonyl group. Okay, so this testosterone has a carbonyl group where the estradiol has a hydroxyl group. Okay. Another difference is that testosterone has an extra methyl group. Okay, so just these additions of these 2 functional groups versus this 1 functional group, it causes big changes in the body, right? So, testosterone, we associate that with the male sex hormone, though, females also produce testosterone just in a lot smaller amount. Whereas estradiol, same thing. Females tend to produce a lot more estradiol than testosterone, and males, they also produce a little bit of estradiol, but not nearly as much as testosterone. So just the fact that we have just these 3 different functional groups can cause huge changes in the structure of, for example, these lions here. Okay, so you can think of testosterone, what that does to the male lion, producing the secondary sex characteristics of the male. Whereas estradiol, female lion produces more of that, and so that helps produce the secondary sex characteristics of that female lion. So functional groups, they can cause big changes in the molecules. Okay. Now that we know about our functional groups, let's talk about how we take a couple of small molecules and we connect them to make a macromolecule. So think macro means large. So large molecules, and those large molecules are actually made up of what we call monomers. So monomers are just subunits of a polymer, a macromolecule. Okay, so you can see an example. Here is an unlinked monomer. So this is some sort of molecule, whatever it is, with a hydrogen on one side and a hydroxyl group on the other side. Now, if we have multiple of these monomers, we can stick them together to form a polymer, a macromolecule. Okay, so polymers, they're large molecules, and they consist of many identical or similar types of building blocks strung together. Okay, and monomers are those building blocks. So we're going to watch a video describing how those polymers come to be using the monomers.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme.
Okay, so from that video we saw actually 2 types of reactions. We saw a dehydration synthesis reaction where we build polymers, and then we also saw the hydrolysis reaction where we break apart the polymers into the monomers. So let's go ahead and talk about those chemical reactions. So let's start out with the dehydration synthesis reaction. So this is where we're making polymers, okay? And this is what we call an anabolic reaction or anabolism, because it requires energy in order to take 2 monomers and bring them together to form a polymer, okay? So think anabolism, it requires energy. And it's to build the polymers. Okay, so how this happens is we have the monomers, and remember the monomers, they have a hydroxyl group on one side and a hydrogen group on the other side. And so when we bring them together, we simply remove the hydroxyl of one monomer and the hydrogen of another, and that forms what? Forms water, okay? And that will allow for that reaction to take place where the 2 monomers will be linked together. So this is an anabolic reaction where we're dehydrating it. We're removing a water molecule to bring 2 monomers together. So we're making polymers. So let me show you what I mean. Here's an example of a short polymer. So remember, polymers are made up of these subunits called monomers. And on one side we have a hydroxyl group, on the other side we have a hydrogen group. And then we have this unlinked monomer over here with its hydrogen and its hydroxyl group. So we're going to undergo a dehydration synthesis reaction where the hydroxyl group and the hydrogen group will break off to form water, okay? And in doing so, that will allow for the short polymer to bond with that unlinked monomer to form a longer polymer, okay? So the dehydration reaction, also known as condensation, you might see in other textbooks, but this reaction is building longer polymers by removing water. Okay. Now the opposite of that is a hydrolysis reaction. So think hydro means water, lysis means breaking, so we're actually going to do the opposite as the dehydration synthesis reaction, where we're going to add water to the polymer to break it apart. Okay, And so since we're breaking apart molecules, in this case, it's a catabolic reaction. So catabolism means that we don't need energy to break the bonds. Okay, so it's we're hydrolyzing it by adding water, and that's going to break apart the polymer into the monomers. Okay. Now, and just, I keep saying that catalysis doesn't require energy. Every reaction does require a little bit of energy, called the activation energy. We'll learn about that later on. So to counter that, we have those enzymes that we saw from that video. Okay, so we have hydrolyze, for example, is an enzyme that speeds up the reaction. Okay, but this reaction would occur where we eventually have the breaking of these bonds. It would just take a long, long time if we didn't have the help of an enzyme. Okay, so we're going to take that long polymer and we're hydrolyzing it, so we're adding water to break the bond. So we're adding water to break the bond. Okay, so now the OH from the water will attach to, in this case, the shorter polymer, and then the H will attach to the unlinked monomer. Okay. So you can see them together. The dehydration reaction is just the opposite reaction as the hydrolysis. So dehydration, we're removing water to basically make a longer polymer, whereas hydrolysis, we're adding water to break the bond, creating smaller polymers, or in this case, a short polymer and an unlinked monomer. So here's a checkpoint question for you. Suppose you eat some cheese. What reactions must occur for the protein of the cheese to be broken down into its amino acid monomers, and then for these monomers to be converted to proteins in your body? So what type of reaction takes place to take the proteins of the cheese and break it down into the monomers? If you said a hydrolysis reaction, I would agree with you. So we're going to add water to break the bonds between the amino acid chains to break them down into individual amino acid monomers. And then that will be small enough in our intestines to absorb those individual little monomers into our bloodstream. And then it can carry those amino acids to our cells, and then our cells can actually build proteins from those monomers, those amino acids. So how do cells build the amino acids into making a protein? That would be through a dehydration reaction, or a dehydration synthesis reaction. Okay, so think hydrolysis, we're breaking larger molecules into smaller, and then dehydration, we're going to build smaller molecules into larger molecules. Okay, so now that we have a basic understanding of organic compounds and the molecules and how molecules are built, we're going to take a look at the 4 major macromolecules of life. So we're going to start out in this video, we're going to learn about the carbohydrates and the lipids. And then in the second video, we'll finish it off with the proteins and the nucleic acids. So we're going to start out with our learning of the macromolecules with our Amoeba Sisters video. Oops, what happened there? It went backwards. Let's try again.
Go ahead and think for a moment about your very favorite food. What is it? Pizza, macaroni and cheese, chicken salad, sushi? Well, we all have different food preferences, but food is a source of large molecules that are needed for life called biomolecules. There are 4 major biomolecules that make up all of life, and this will be the focus of this video. Before we get into details about the 4 biomolecules, we need to talk about one very important vocabulary word, the word monomer. a monomer is a building block. If I had some large substance, the parts that make up the substance are called monomers, just like building blocks. We're going to talk a lot about monomers today because we need to understand what the biomolecules are made of. And we need to understand biomolecules because they're building components of life. So let's introduce the 4 biomolecules now and talk a little bit about their functions. We'll start with carbohydrates, carbs. Well, carbs are something that you've probably heard a lot about when people are talking about diets. You know, they try to go low carb, or maybe they want a lot of carbs. Diets always come and go. Pasta and breads are examples of foods heavy in carbohydrates. Carbs are actually a very important source of energy. In fact, that's one big function of carbs. They are a great, fast source of energy. If you are a marathon runner, you might want to eat a lot of carbs the night before a race. Lots of marathon runners do this. It's called pasta loading. They eat a big pasta dinner the night before they go out on their marathon. Now, carbs have a monomer. Again, remember, monomers are building blocks. The monomer for a carb is a monosaccharide. I know that's a big mouthful, but monosaccharides make up carbohydrates. Next one up is a diverse group known as lipids. Lipids are better known as fats, and they have 2 different types of building blocks. One type of building block is called a fatty acid, and the other type is called a glycerol. Now, examples of lipids include butter, oil, and cholesterol. Lipids, though, they have a lot of great functions. You might think, well, that's fat. How good can fat be? Well, it just depends when you put it into context. For example, you know those really adorable seals that you see on calendars? They have this fluffy white hair. They're actually called a harp seal. Well, they only look like that when they're babies. When they get older, they're not quite as cute. In their little baby stage, they actually have a lot of this hair that they're born with that keeps them warm. But over time, they have to develop blubber. It's fat, and it helps keep them warm. Lipids are great for insulating. Also, you might not think about fats as being related to energy, but fats are a great source of long-term energy. They can store energy for a long time. Say, for example, you wanted to swim the English Channel. That's like 21 miles of swimming. The fastest swimmers might be able to do that in 7 or 8 hours, but it might take a lot longer than that for the average swimmer. More like 25 hours, and that's a lot of swimming. Well, you would want to make sure that your body has enough lipids, enough stored fat that it can pull upon. Because after you burn off those carbohydrates, remember, carbs are the fast source of energy, well, you might not have enough energy storage unless you have some lipids on hand. Lipids also make up cell membranes, so they are very important for life because all living things are made of cells. Of course, an excessive amount of lipids could be a bad thing for your health. Remember, it's all about moderation. Okay, next, proteins. Now, when you hear about proteins, a lot of times you might think about power bars. They say they have a lot of protein in them and that they help with muscle building. Well, protein is great for muscle building. Examples of foods that are high in protein include meats and many types of beans. The monomers of protein are amino acids. So sometimes you see these labels that say, this has 20 amino acids in this food. And really, they're just trying to say that it has protein. Because proteins are made of amino acids, so that's just some fancy advertising for you. But in addition to being important for muscle development, protein is also very important for other functions, such as working in the immune system and acting as enzymes. Remember, enzymes are made of proteins, so proteins are important for the body. Now, when we start talking about genes, the DNA genes, not the genes you wear, the DNA codes for proteins that are very important for structure and function in the body. The last biomolecule is known as a nucleic acid. Nucleic acids include DNA and RNA, which we'll get to when we get to genetics. They have a monomer called a nucleotide. It's going to be an easy one for you to remember because nucleotide sounds a lot like nucleic acid. And if considering DNA and RNA, both of these are involved in genetic information for the coding of your traits. They are found in a lot of your food because whenever you eat something that came from something once living, it can still contain the DNA. For example, when you eat a strawberry, you're actually consuming all the cells that make up that strawberry. And in the nucleus of all those strawberry cells is DNA. Plants and animals both have DNA. In fact, any type of life must contain nucleic acids to direct the cell's activities. So we just powered through introducing the 4 biomolecules by providing examples, exploring their monomers, and giving some general functions. One last very important part to mention is the structure of these biomolecules. Understanding the structure can help with predicting their properties and easily being able to identify them. One thing I like to tell students to do is to write the 4 biomolecules in the same order that we went through: carbs, lipids, proteins, and nucleic acids. Then remember this mnemonic device that goes with these 4 biomolecules: cho, cho, chom, chomp. Instead of chomp at the end with an M, it's chomp at the end with an N. The C stands for carbon, the H stands for hydrogen, the O for oxygen. So carbs, lipids, proteins, and nucleic acids all have that CHO in there. It's just that proteins and nucleic acids also have an N, which is nitrogen. And nucleic acids additionally have a P, which is for phosphorus. So again, CHO, CHO, CHOM, CHOMP, the major elements in the 4 biomolecules. Now, these elements are arranged differently in the 4 biomolecules. It's important to explore the arrangement of the elements in biomolecules because the structure of that arrangement greatly impacts the biomolecule function. So, to the Google to discover some biomolecule arrangement illustrations. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so from that video we got a great introduction into the 4 macromolecules of life. So in this video we're only going to cover the first 2, the carbohydrates and the lipids. And as we go through, we're going to learn about the structure as well as the function of the macromolecules. So let's start out with the carbohydrates. So the carbohydrates, if we just look at its monomer, okay, the smallest unit, we call those the simple sugars, otherwise known as the monosaccharides. Okay, so monosaccharides are the monomers of our macromolecule carbohydrate. Okay, but when we add in multiple monosaccharides together, we will form a polysaccharide. So think poly means many, saccharides means sugar. So polysaccharides versus mono means one, so monosaccharide would just be the monomer. So multiple monosaccharides together form a polysaccharide. Okay. So let's take a closer look at the structure. Now if we take a look at this, the structure of our carbohydrate, you'll notice again we have our carbon skeleton. And the carbon skeletons of these monosaccharides, or the monomers, they can range anywhere from 3 carbons to 7 carbons long. So in this case, we have a 6 carbon sugar molecule here. Since it's 6, it will form a ring. And in this case, since it's 6, it forms a hexagon. So it's also known as a hexose. Okay, so the 3 to 7 carbon structures, the 3s and the 4s and the 7s are more rare, but you'll see more monosaccharides with 5 carbons. We call those the pentoses, and the 6 carbons the hexoses. Okay. So, like I said before, the monomer is the monosaccharides. So mono means one, saccharides means like sweet or sugar, and so we just have one single sugar there. Now what's one interesting thing about our carbohydrates, particularly our monomers, is that they all follow the same ratio of carbons to hydrogens to oxygens. So you can see here we have a molecular formula where we have 1 carbon to 2 hydrogens to 1 oxygen. So this is going to be pretty standard with our monosaccharides, okay? So if we take a look at our 2 examples here, we have glucose and fructose. Those are both types of carbohydrates, and they both follow that same ratio, where we have twice as many hydrogens to carbons and oxygens. So both of them has 6 carbons, 12 hydrogens, and 6 oxygens. So it follows in that same ratio where we have twice as many hydrogens to carbons and oxygens. Now, remember these 2 particular molecules, they have the same chemical formula, but different structural formulas or structural placements there. And so these, of course, are examples of isomers. Okay, so the glucose and a fructose is an isomer. And in this case, what determines their function is the placement of that functional group. What was it called when you have carbon double bonded to oxygen? That's a carbonyl group. So the carbonyl group, if it's in the second carbon, that would be fructose, versus in the first carbon, that's glucose. Okay. And then we can see some other examples of monosaccharides besides the glucoses and the fructoses. We have galactose. Galactose is another type of monosaccharide, and ribose. Ribose is an example of a pentose that has 5 carbons, and we'll learn more about that when we talk about our nucleic acids. Okay, so here is a checkpoint question for you. Go ahead and write the formula for a monosaccharide that has 3 carbons. Okay. So remember, with our monosaccharides, they fall under the same, general ratio of carbons to hydrogens to oxygens. So if we have 3 carbons, how many hydrogens will we have? So we have twice as many, so that would be 6 hydrogens, and then 3 oxygens. So it would be C3H6O3 would be the chemical formula. All right, so let's take a closer look. Let me go back. Okay, so that was our checkpoint question. Let's get into the polymers now. So we talked about how we have our monomers. In this case, we have an example of glucose. So if we take 2 glucose monosaccharides and they undergo a dehydration synthesis reaction where we remove the water molecule from them, they will form a polymer. In this case, it's called a disaccharide. So di means 2, saccharide again means sweet or sugar. So in this case, we have 2 simple sugars linked together. And this is a covalent bond like any of the other ones. But this is because it's a carbohydrate, we call it a glycosidic linkage. So it's just another name for a covalent bond. In this case, it's the covalent bond of carbohydrates. So we have glucose and glucose held together to form a disaccharide. And this particular disaccharide is called maltose. Okay. Here's another example of a dehydration synthesis reaction. In this case, we have glucose and fructose. So when we bring those 2 molecules together with a dehydration synthesis reaction, forming that glycosidic linkage, we get the disaccharide called sucrose. And you may be familiar with sucrose, that's table sugar. So let's watch an animation showing how we bring together those monosaccharides to form the disaccharide.
Sometimes organisms link sugar molecules in pairs to form disaccharides. Here are several examples. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugar cane and sugar beets and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of two linked glucose molecules. Digestion of starch in a sprouting seed or in the intestine of an animal produces this disaccharide.
Okay, so we can see how those disaccharides are formed from just simply linking together different monosaccharides. All right, let's do a checkpoint question. So we have lactose. Lactose is the disaccharide sugar in milk. It is formed from glucose and galactose. The formula for both of these monomers is C6H12O6. So what would be the formula for lactose then? So if we're bringing together two monomers, both of them have 6 carbons, 12 hydrogens, and 6 oxygens. So first of all, what type of reaction is that? That would be an example of a dehydration synthesis reaction. And in order for that to occur, water must be removed. So we simply need to double the number of carbons, hydrogens, and oxygens and subtract a water molecule. So lactose must be 612H, not 24, but 22 because of water. And oxygen, instead of having 12 like carbon, it would have 11. Okay, so the chemical formula for lactose is C12H22O11. Okay, plus we'll end up with a water molecule, H2O. Okay, so let's take a look at our final part of the carbohydrate, the polymer, which we call the polysaccharides. So polysaccharides mean many sugars or many saccharides linked together. So with this we have basically 3 main types that we go over. We have starch, glycogen, and our cellulose. Now cellulose and starch are found in plants. Okay, so plants, they have these two polysaccharides. Animals, we have glycogen. And if you take a look at the structure, all 3 of them are made of the same monomers. They're all made of glucose. It's just the arrangement of the glucose. Okay, so you can see how starch, its arrangement is slightly different from glycogen. Glycogen, it kind of branches off, whereas starch is more linear. And then cellulose, we have a more organized structure to it. Okay? So if we think about starch, what plant products are high in starch? You know, think of potatoes, pastas, things like that. So the starch molecules of plants, those are a food storage for plants. Okay, since it's just a bunch of glucose linked together, it can be stored away inside the plant as a food storage, because glucose is a very, very important molecule for energy. So starch can store the glucose away in a longer polysaccharide molecule. Okay, glycogen is basically the same thing for us. Okay, so it's like starch for us. So its monomer is still glucose, and it's a glucose storage for animals. And you can see that it's highly branched compared to the starch. Now, where we tend to store glycogen tends to be in our muscle cells, because if you think about it, muscles, they need lots of glucose to undergo chemical reactions to move. And so muscle cells, as well as in the liver, those are 2 big organs, or muscles are, you know, an organ system, muscular system. that is good at storing glucose as polysaccharide glycogen. And then finally we have cellulose. So cellulose is, it doesn't have anything to do with energy. Instead, the cellulose, it makes up the cell walls of plants. Okay, so remember that animals, we don't have cell walls, but plants, they have cell walls surrounding their cell membranes. And these cell walls are made of cellulose. And cellulose happens to be the most abundant organic compound on Earth, okay, because we have lots of plants out there. And so plants are at the bottom of the food chain, so there's a lot of plants out there. Each plant is made of, millions of plant cells. Each plant cell has a cell wall made of cellulose. Okay. We, in our diet, we can certainly eat starch, but we cannot actually process cellulose. So when we eat, things that are high in cellulose, basically think of plant parts, they tend to be indigestible by us. And so this is what we call dietary fiber. Okay, so if you eat lots of, lettuce, lettuce is just plant leaves, right? And it has lots of cell walls from plants, and so that is our insoluble fiber, so something we can't really break down in our bodies. We don't have the enzymes, at least we don't. Some animals have gut bacteria that will help break it down for them. Okay. And then there's one more type of polysaccharide you might see mentioned in your textbook. It's called chitin. So chitin is basically, it's similar to a cell wall of a plant, but it's used in the exoskeleton of some of our invertebrates, such as our insects and our crustaceans. So it helps build the exoskeleton. Chitin is also the cell wall in the kingdom fungi. Okay, so instead of having cellulose as their cell wall, they have chitin. So just another example of a polysaccharide. Okay, so let's watch a video on polysaccharides.
Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides.
Okay, so that was short and brief and to the point. So you can see here we have our three main polysaccharides: starch, glycogen, and cellulose. And they look very, very similar because they're made of the same monomer, the glucose. And you can see some slight differences. Okay, so for example, look at starch compared to cellulose. Okay, so starch, we have, opposite sides, we have the same side for the CH2OH, whereas in cellulose it's opposite sides. Okay. We talked about how starch and glycogen are very, very similar. The only thing is that glycogen tends to be branched. Okay, so again, here's just showing the difference between starch and cellulose, which both plants produce. And in this case, we have starch, the OH is highlighting being on the same side. Okay, so we call that an alpha glucose instead of opposite sides, the beta glucose. Okay, so cellulose is just slightly different structural isomer, you can see, compared to starch. Now, what are the functions of carbohydrates? Okay, so why do we need carbohydrates in our bodies? Why do we consume them in our diet? And you can think of, like, what is it, 45% of our diet is carbohydrates, something like that. Well, because they're our energy source. So saccharides, sugars, they are quick energy for our bodies. Okay, so we need that glucose. It's used in a process known as cellular respiration, which we'll talk about later in, I believe, chapter 7. And cellular respiration is that process where our mitochondria, they take glucose, that organic molecule, and they can convert it into a usable form of energy for the cells called ATP. So we'll talk more about that in chapter 7. But for now, we just need to know that carbohydrates, they are our main form of energy for our bodies. Okay? They also are structural, so think of the cellulose of the plant cell walls. Okay, so cellulose is used to create that rigid structure of the cell wall of a plant. Okay, so there is a structural component of our carbohydrates. So that's it for the carbohydrates. So now we're going to switch over into the lipids. So the first thing I think of when I think of lipids are fats. But it's actually a very diverse group of macromolecules. In fact, it's so diverse that there's no true polymers or monomers. Okay, so it's very, very diverse. You can't, have monosaccharides making polysaccharides. You can't have, a monomer lipid making a polymer lipid because they're very diverse. So what brings them together in their diversity is that they are hydrophobic. Okay, so that term hydrophobic, hydro means water, phobic means fearing. So they tend to stay clear of water, right? So think of, lipids being fats. If we take something like oil, which is, made of fats, and combine it with water, what happens? You get a layer, right? So the oil does not want to mix with the water. And so it's hydrophobic. And the reason why it's hydrophobic is because it contains mostly hydrocarbons. And if you remember, hydrocarbons are just basically, carbons linked together with a bunch of hydrogens surrounding them. And because of that, they are nonpolar. Okay, so nonpolar does not mix with polar, our water molecules. So lipids tend to be nonpolar and therefore hydrophobic. Okay. So we're going to take a look at 3 main categories of lipids. We're going to talk about the fats, the phospholipids, and the steroids. So the fats, those are basically made of 2 components. We have a glycerol group and a fatty acid. Okay, so again, there's no real monomers. Okay, so we have glycerol and fatty acids that makes up a fat, okay, which would be the lipid. Phospholipids, they also have fatty acids like fats, but they also have a phosphate group. What's unique about that is phosphate group is polar, whereas the fatty acids are nonpolar. Okay, so we'll talk more about that when we get into our cell membranes, because they make up our cell membranes. And then finally we have our steroids. So steroids, they actually have carbon rings. So we have 4 carbon fused rings. And so again, it's very different from a fatty acid or a phospholipid or a glycerol. So you can see the diversity with our lipid macromolecule group. So we're going to watch a video. So that video kind of describes them.
Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long nonpolar hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules. The fatty acid tails in a fat can vary in length. A very important characteristic of fats is the number of double bonds in the hydrocarbon tails. Fats that contain only single bonds are called saturated fats. Fats containing double bonds in 1 or more tails are called unsaturated fats. Unsaturated fats tend to be liquid at room temperature. Vegetable oils are examples. Saturated fats, like butter and lard, are solid at room temperature. Saturated fats in the diet can lead to heart disease, while unsaturated fats are safer.
Okay, so we got a good introduction into those fatty acids, otherwise known as triglycerides. So you can see at the beginning of the film, it showed you that dehydration synthesis reaction where we took our glycerol molecule and removed a water molecule to bring the fatty acids. Okay, so here's that dehydration synthesis reaction for a glycerol molecule and a fatty acid. Okay, and so that's bringing that in. Now, when we have that type of dehydration synthesis reaction, we form a covalent bond, just like with the carbohydrates. But instead of a glycosidic linkage, because this is a lipid, we call it an ester linkage. So again, ester linkages, they are just, the covalent bonds for lipids, whereas the glycosidic linkage is the covalent bonds for carbohydrates. Okay. So in this case, you can see that the fatty acids, they're basically just carbons and hydrogens. So those are the hydrocarbons. And so because they are nonpolar, they're going to not want to mix with water. So they are hydrophobic. Now, if we take a closer look from the video, it talked about the difference between saturated and unsaturated fats. So saturated fats and unsaturated fats, they're both made of glycerol with the fatty acid chains. The only real difference is how many hydrogens make up the fatty acid chains. So if it's saturated with hydrogens, like let me go back a slide. If it's saturated with hydrogens, meaning we have, carbons single bonded to other carbons and then single bonded to hydrogens, there's no room for any additional hydrogens here. So it's saturated with hydrogen. So this is an example of a saturated fat. An unsaturated fat is where we lose hydrogens because we have the carbons double bonding together. And since they're sharing, two sets of electrons, they don't need that extra hydrogen, okay, to bond to. And so that creates a little kink in the chain if you see here. And because of that kink in the chain, you can see there's more space between the fatty acids. And so when we have these kinks in the chain, and again, it kind of depends on how many kinks there are, it will lead to a more liquid structure. Okay, so these fatty acids, since there's more space between them, they're going to be liquid at room temperature. Whereas when they're saturated full of hydrogens, then it's going to be a solid at room temperature. So our animal-based fats are high in saturated fat, whereas our plant-based fats are high in unsaturated fats. Okay, the second group that we're talking about are the phospholipids. So the phospholipids, actually let me go back. So we talked about the saturated and unsaturated fats. Let me go to the trans fats. So trans fats, you've probably heard of, and you want to stay away from them. So basically what happened is, the food industry, they decided that they wanted to take plant-based fats and turn them solid at room temperature in order to get, a different, instead of using saturated fats, which were mostly animal-based. And plant-based fats tend to be cheaper to produce because they come from plants, whereas saturated fats, are animal-based, and so they come from animals. So it's a lot easier to raise a bunch of plants than it is to raise a bunch of animals. And so the food industry took a hold of that and tried to convert our plant-based fats into a solid form. And so by the 1990s, we had a lot of these partially hydrogenated oils. And you can see there's actually not much difference. So here's just a regular unsaturated fat. And you can see that it's we have our double bond that makes it unsaturated, creating that kink. And that kink leads to the more liquid at room temperature. But if we change the structure just by simply balancing it out, where we have a hydrogen on one side and a hydrogen on the other, we get a trans fat. So think trans, opposite sides for the hydrogens. Cis is same side. So this is the normal unsaturated fat. This is the trans fat. So this was produced by the food industry. And notice, because it's the hydrogens are on opposite sides, they don't create that kink, and so they tend to be solid at room temperature. The problem, though, is that our body does not know what to do with this. Okay, so it's used to dealing with the unsaturated fats as well as the saturated fats. But when you get something like that, we don't have enzymes to break this apart. Okay, so each polysaccharide or lipid or protein has enzymes that can break it down into its monomers or into smaller molecules. But trans fats, we don't have those enzymes. Okay, our body can't recognize this, so we don't have special enzymes to break those bonds. And so they tend to accumulate in our body and cause a greater health risk than, say, a saturated fat. So you can see, the health risk here. If we stick to unsaturated fats, there's different types. Like a monounsaturated means that there's one double bond. Unsaturated means multiple double bonds. So they tend to be, less risk to our health. But saturated fats, because they have, more hydrogens, we want to eat them in moderation because they do pose some risk. Trans fats, because our body simply doesn't know what to do with them, they pose the greatest risk. Okay, so trans fats are not good. Luckily, the government has figured that out through countless studies and has banned them. So we still have some products on the shelves that still contain trans fats, just simply because they tend to be in highly processed foods, and highly processed foods have a long shelf life. But they were banned. I think food industry could no longer use them after, I think it was like 2018. But you still might see them on the store shelves for those products that can last a long time because they are heavily processed. All right, so now that we talked about the fats and the trans fats, saturated fats, and unsaturated fats, let's go back to that phospholipid slide. So the phospholipids are another category of lipids, and as I stated before, there's something unique about the phospholipids. They actually have a lipid component, being the fatty acids, which are hydrophobic, but they also have a phosphate group. Okay, and if you recall from that phosphate group, that was one of our functional groups, and that functional group is hydrophilic. So we actually have a hydrophilic component to our phospholipid. So this makes a kind of a unique situation here. These phospholipids are found in our cell membrane, so it's what makes up the majority of our cell membrane. And our cell membrane is what's called a phospholipid bilayer. So we actually have two layers of lipids, phospholipids, and you can see how they're arranged. So we have the phospholipid head, which contains the phosphate group sticking outward, and then the hydrophobic fatty acid tails facing inward towards each other. So this creates that barrier. So you have kind of this hydrophobic area facing inward, and then outside you have the hydrophilic heads. Okay, so this is kind of a unique type of lipid that contains both a hydrophilic part and a hydrophobic part. And then finally, we talk about steroids. So we did a little bit of steroids already in when we were talking about the functional groups. So for example, the sex hormones, testosterone and estradiol, those are basically formed from a steroid called cholesterol, and you're probably familiar with cholesterol. So that is a lipid based structure. And if you recall with I should have put another example of it, but the testosterone and estradiol, remember that it had those rings. So steroids have those ring structures. We, of course, as humans, we produce testosterone. Males, of course, produce more testosterone than females. And what happens to males when they produce testosterone? It allows them to get the secondary sex characteristics of males, such as larger muscles. And so, of course, the fitness industry have figured that out and have synthesized the male sex hormone testosterone to form a variant which we call an anabolic steroid. So anabolic steroids, what they do is that they mimic testosterone in the body, which helps men produce muscles. And so if you're injecting anabolic steroids into your body, you are able to make muscles very quickly. The only problem, though, is that since the muscles are produced pretty quickly, they tend to be more fragile. So people that take anabolic steroids, they tend to get injured a lot more than people that are just doing it the natural way. Also, there's other consequences when you're adding a synthetic hormone into your body. It can definitely screw up your other hormones and cause other issues with your body. So it's not a good idea to do that. So it can cause weaker muscles that appear bigger, which can lead to more injury as well as some other issues. Because basically, if you're injecting yourself with synthetic testosterone, your real testosterone will stop being produced. And so that's not a good thing. Anyway, checkpoint question. So explain why fats and steroids, which are structurally very different, are both classified as lipids. So remember, steroids are ring-shaped structures. They're not like those carbon skeletons like fatty acids. So why are they both classified as lipids? Well, if you recall, the lipids are classified based on them being hydrophobic. So steroids are hydrophobic, so are our fatty acids or fats. And so therefore they're categorized in the same category as a lipid. Okay. Also, if you recall from our Amoeba Sisters video, they're made of those three elements. Okay, so they're a CHO, just like carbohydrates being a CHO. They contain carbons, hydrogens, and oxygens. So what are the functions of lipids? Well, the video alluded to the fact that they are also used as energy. Your body will use carbohydrates first, okay, but then it will also rely on lipids as a fat storage for energy. So if you think about it, we have 3 meals a day, but our body is constantly burning sugar, burning energy throughout the day into the night. And so, eventually our body kind of loses the amount of glucose that have accumulated from our 3 meals a day plus snacks. But in the middle of the night, you still need your glucose. Okay, so lipids can be used in replacement of that. So that's why we continue to have lipids in our diet as an energy source. They also form protective functions. So think about in your body, we have layers of lipids that surround our vital organs. So that helps with cushioning and protecting our vital organs, so that if you're running or doing jumping jacks, your liver is not slamming into your stomach or something like that. That could cause damage. So the protective layer of that fat can help. And then finally, it's also involved with structural components. So the previous video alluded to the fact that our cell membranes are made of phospholipids, and since phospholipids are a type of lipid, our cell membrane is made of lipids, okay? And that creates separates the inside of the cell from the outside of the cell. So all of our cells have that. All right, so let's do a concept check question to make sure we're understanding our carbs and our lipids. So all lipids, A, are made from glycerol and fatty acids, B, contain nitrogen, C, have low energy content, D, are acidic when mixed with water, or E, are poorly water soluble. So what unites all lipids? If you chose E, I would agree with you. They are poorly water soluble because they're hydrophilic. Okay, so our lipids do not mix well with water. So that's the reason why they're classified as lipids. Now we do have some lipids that are made of glycerol and fatty acids, such as our saturated and unsaturated fats, but not all lipids, like our phospholipids and our steroids. They don't contain nitrogen because that would be a CHON instead of a CHO, so that's incorrect. They do have high energy content, okay, so fats can be burned to give us energy, okay, and they're definitely not mixing with water to form an acid, so E is the correct answer. Here's another question for you. The formation of starch from simple sugars such as glucose involves a series of what type of reactions? Would that be a hydrolysis reaction, dehydration reactions, hydrophobic reactions, or denaturation reactions? So what type of reactions form the starch? So if we're forming a polymer from monomers, we undergo dehydration reactions. Okay, so we take the monomers, take out a water molecule to link them together, so we're dehydrating them to form the bigger molecules, the starch. So B is the correct answer. All right, so by now you should be able to describe the importance of carbon to life's molecular diversity. So think about, organic compounds. You should be able to describe those chemical groups that are important to life, such as the hydroxyl, the carbonyl, the carboxyl, the amino, and the phosphate. And then sometimes you even see the sulfohydryl, but we didn't go into that one, as well as the methyl. And then you should be able to define monosaccharides and how you can make them into disaccharides and then into polysaccharides. And then give some examples as well as explain their functions. Okay? And then define lipids. Okay, so what makes a lipid, as well as what distinguishes fats from phospholipids and steroids, and explain their functions. And then finally, you should be able to explain how trans fats are formed in food, as well as describe the evidence that suggests that eating trans fats is more unhealthy than consuming saturated fats. So all of these objectives we covered in this video. So for the next video, we're going to finish off the chapter by looking at the proteins and the nucleic acids, our last of the 4 macromolecules of life.